Welcome to the Deep Dive. Today, we are opening up a really remarkable stack of sources. They challenge pretty much everything we think we know about innovation and, well, access. We're diving into the system called QuantumCURE. It's a drug discovery pipeline, but it merges artificial intelligence, huge public data sets, and, yeah, cutting-edge quantum technology.
And the truly astonishing part, the headline here is, who built it? This wasn't some billion-dollar pharma lab, or a massively funded startup. No, it was built by an independent inventor, a retired software engineer, Mansour Ansari, basically working out of his back office in Oklahoma.
That's the core story that makes this deep dive so fascinating, right? Our mission today is really to explore how one person, determined, could assemble a pipeline that genuinely rivals the big industry players. The sources are pretty clear on this. This whole effort proves, and I'm quoting here, one person with persistence and imagination can change the course of science.
Exactly. And it really came out of a clear need addressing the problem.
Right.
Current drug discovery is just so slow. It's exclusive, incredibly expensive. We're talking, 10, 15 years? Billions of dollars often just for one single drug to make it to market.
Right. And so the solution QuantumCURE puts forward is essentially democratizing that whole process. It builds tools that integrate, the classical world, chemical databases, AI, with the unpredictable nature of quantum randomness, high entropy physics. So the core takeaway for you listening should be the sense of pure possibility, that the frontier, it's actually closer, much more accessible than maybe the big institutions want us to believe.
Precisely. It's really a testament to the power of open source knowledge, modern computation. Innovation isn't necessarily locked behind those huge proprietary walls anymore.
Okay, let's unpack this then. Before we get into the quantum side, the sort of bells and whistles, we need to understand the foundation, the accessible classical scaffolding. I mean, this field has a long history, right? From ancient healers chewing willow bark through industrial synthesis, and then really taking off in the digital age, maybe the 80s, 90s, with computational chemistry.
That's right. That's when we started modeling molecules digitally. And the inventor's starting point was basically leveraging these modern, publicly available tools. Anyone can use them to set up a pretty high-quality baseline discovery system.
So the barrier isn't really access to the knowledge or the basic tools anymore. It's more about persistence and knowing how to put the pieces together. That changes everything. Okay, let's look at the toolkit. The sources laid out pretty clearly.
Right. It starts with the data. You need three massive engines. First, there's the knowledge engine, PubMed. That's the U.S. National Library of Medicine's archive. Over 38 million biomedical papers. You just can't start without knowing your target.
Okay, PubMed for the background. Then you need the actual ingredients, the chemicals.
Exactly. That's PubChem, hosted by the NIH. It's like the chemical universe, over 118 million compounds.
Yeah.
That's your, potentially infinite starting library.
Wow. And third, how do you connect those chemicals to what they actually do in the body?
That's the critical link. ChEMBL. It's a bioactivity database. It connects molecules to known experimental results, things like binding affinity, toxicity data, super important.
So that's the data spec. Huge amounts of information. But you can't just stare at lists of chemicals, right? You need software to actually work with them, test them virtually.
Right. And this brings us to the essential open source software layer. The big one here is RDKit. People often call it the Python Swiss Army knife for cheminformatics. It's fundamental because chemicals are often just written as text strings, smiles notations. RDKit takes that flat text and turns it into a usable 3D molecular structure. You absolutely need that for simulations.
Okay, so it makes the molecule real for the computer. What else does it do? You mentioned properties, like log P. Why is that so important?
Log P, yeah. It tells you the molecule's partition coefficient. Basically, is it more fat-soluble or water-soluble? This is just critical. For a drug to work, it usually has to cross cell membranes, right? If it's too water-soluble, it might bounce off. Too fat-soluble, maybe it gets stuck in fatty tissues and never reaches the target. RDKit calculates this complex property instantly.
Makes sense. So RDKit prepares the molecule. Then what's the main event, the virtual testing part? You mentioned AutoDock Vina.
Yeah, Vina is the docking workhorse.
Yeah.
It does the molecular docking. It predicts how a small molecule, the potential drug, might bind to the active site of a big protein target. And crucially, it estimates the binding affinity, how strongly it sticks. A lower score, a lower energy, means a better potential fit.
So if you're determined, you grab PubMed, PubChem, ChEMBL data, use RDKit for prep, Vina for docking, put it all on a decent computer. And you've basically built yourself a professional-grade classical discovery platform. The baseline is set.
Exactly. And that baseline itself proves a huge point. It was the biggest barrier in the past, just access to the tools. That's pretty much gone now.
Okay, now this is where it gets really interesting for me. If these classical simulation tools are already so good, running millions of calculations, why bother moving beyond them? Why bring in quantum hardware? Seems like a lot of extra complexity, a computational overhead.
Yeah, it's a fair question. It really boils down to a fundamental limit of classical computers, how they handle randomness. Classical simulations use what are called pseudo-random number generators, PRNGs. They're fast, efficient, but they're algorithms. Mathematical recipes, which means ultimately they're deterministic. If you know the starting point, the seed, you know the entire sequence of number that will generate.
Right. And in this absolutely massive search space, all the possible ways a molecule could fold or dock those deterministic sequences could create blind spots. Like they might repeatedly explore some areas but completely miss others.
That's the core problem quantum mechanics helps solve. The quantum turn here involves injecting QRNG, quantum random number generators. This isn't an algorithm. It's true randomness. It's harvested from actual physical quantum processes, the inherent entropy, the unpredictability at the quantum level. This can push the search into totally new directions, places a PRNG might literally never stumble upon because its algorithm doesn't lead there.
And the sources show this isn't some unobtainable lab setup. The inventor used cloud access to commercial quantum hardware, right?
Precisely. We're talking about access to specialized machines like D-Wave quantum annealers. These are really good at optimization problems, which is perfect for navigating those complex energy landscapes in molecular docking.
And also things like IonQ's gate-based quantum computers, which are maybe more general purpose.
Yeah, more for universal quantum algorithms, maybe for more detailed quantum chemistry calculations down the road. The key point isn't replacing classical methods entirely. It's about augmenting them, adding something new.
So the main goal is expanding the search space, using that quantum randomness to find things the old way might miss.
Exactly, expanding the search space. A quantum-seeded system might sample rare molecular shapes or find really unexpected novel ways a molecule could dock that the PRNG, being deterministic, just overlooks time and time again.
So it's a sophisticated hybrid approach then. Use PRNG for the fast baseline stuff, inject QRNG to find new paths, maybe use annealing to really zero in on promising areas.
That's the idea. Combining the strengths of each approach.
Okay, let's look at the actual pipeline architecture. The simulation's job is to answer that basic question. Does this molecule bind to this protein in a way that could actually affect a disease? The sources use a classic example, EGFR, the epidermal growth factor receptor, big cancer target. The protein structure is defined by its PDB ID, 1M17.
Right, PDB ID 1M17 is just the specific 3D map, the coordinates of that EGFR protein structure. Vina needs that map to do its calculations. And the workflow is pretty rigorous. You start with protein preparation, basically cleaning up the 1M17 structure, then ligand preparation, taking the SMILE string from PubChem, turning it into 3D with RDKit, then the docking itself using Vina, and finally post-processing, analyzing the results, ranking them.
But here's the crucial part for the quantum angle. The quantum seeding requirement. Every single Vina run needs a random seed number to kick off its sampling. And this system insists that the 64-bit seed, which is pretty standard for security, right, has to be logged every time.
Yes, and not just the seed itself. The log has to include the provenance, the source tag. Was this seed generated by a local USB QRNG device, a cloud-based QRNG service? Or if those failed for some reason, did it have to fall back to a PRNG? This meticulous logging is absolutely critical for quality assurance, reproducibility, making sure you know exactly how each result was generated.
Okay, but I have to push back a bit here. This logging, the extra admin, dealing with external quantum services, is the overhead of using QRNG really worth it compared to just using a standard, high-quality PRNG? Doesn't it just slow things down?
That's the million-dollar question, isn't it? And it's exactly what the inventor wanted to test. The data coming out of the system suggests, yes, it is worth it, for maybe two main reasons. First, reproducibility and quality. The inventor comes from software engineering, right? So this logging is like applying a rigorous QA mindset, maybe something traditional wet labs sometimes, gloss over. It ensures you can trace everything.
Okay, QA makes sense. What's the second reason?
Novelty, plain and simple. When they run A/B comparisons, same ligands, same protein target, one run seeded with PRNG, one with QRNG. QRNG runs consistently show, well, the significant shifts in the docking scores. And maybe more importantly, higher pose diversity. It means the quantum randomness is genuinely nudging the simulation into exploring different binding configurations, ones the PRNG tends to ignore or miss.
So the real power isn't just finding one score that's slightly better. It's the potential to consistently uncover new ways molecules can bind, things that maybe haven't been patented yet, finding novel intellectual property.
Exactly. That potential for novel IP discovery, finding truly new chemical space. That's what could justify the extra overhead entirely.
Okay, so you've run thousands, maybe hundreds of thousands of these quantum-seeded docking simulations. You've got this mountain of data. How on earth do you find the few potential winners, the needles in that giant computational haystack? This has to be where the AI comes in, right? The intelligence layer.
Absolutely. AI acts as the essential filter, the translator. Its core job is decision-making at scale. It has to sift through all that output, filter it, rank it, and ultimately narrow it down from, say, hundreds of thousands of DOC compounds to a really high-confidence top 10 shortlist. The sources call this the wet list, the ones you'd actually spend money synthesizing and testing in a real lab.
Now, the sources also outline this really interesting strategic shift plan for around 2026, moving from just looking at the docking to looking at the biology first. Multi-omics integration. So docking tells us if a molecule could bind, but multi-omics answers the bigger question: should we even be targeting this protein? Is it biologically relevant to the disease?
Yes. And if you connect this to the bigger picture, this is where the system starts to listen to the whole living orchestra of biology, as the source puts it. Docking alone can give you a lot of false positives. A molecule might look great in silico, perfect binding score, but it does nothing useful, or maybe it's toxic in a living system. Multi-omics layers in that biological context, that necessity to reduce that risk.
Okay, let's break down that orchestra. The sources mention 4 key omics layers. First up, transcriptomics.
Transcriptomics looks at the cell's mRNA. You can think of it as the cell's activity log. It shows which genes are currently active, being transcribed into instructions. This tells you which biological pathways are busy or maybe hyperactive in a disease state.
Okay, the activity log, then proteomics.
Right. If transcriptomics is the instruction manual, proteomics is looking at the actual machines being built and running. It measures the proteins themselves, and proteins are what drugs actually interact with most of the time. You might see a gene being transcribed, high mRNA, but if the protein isn't actually being made or is quickly degraded, targeting it won't work. Proteomics checks that.
Got it. Third layer, metabolomics.
Metabolomics tracks the small molecules, the fuel, and the waste products in the cell. It's like checking the exhaust fumes or the supply chain of a factory. Often the changes in these metabolites, the echoes of pathway activity, can reveal disease vulnerabilities or confirm that a pathway you want to target is indeed active.
And finally, epigenomics. This sounds different.
It is. Epigenomics is often called the software layer on top of the DNA hardware. It involves things like DNA methylation or histone modification that control how genes are read, without changing the DNA sequence itself. Think of them as switches. Sometimes targeting an epigenetic factor could flip the switch on an entire disease pathway, turning it off.
Wow, so the goal is to integrate all 4. Looking for agreement, concordance.
Exactly, concordance. If the transcriptomics says a pathway is hot, the proteomics shows the key proteins are present, and the metabolomics confirms the pathway's activity. Then targeting a protein in that pathway becomes a much higher priority. The AI can then use techniques like signature reversal, basically, finding drugs whose effects oppose the overall disease signature seen across these omics layers. Great for drug repurposing, too.
And to make this whole system really convincing, pharma credible, the inventor added some specific enhancements.
Yes, several important ones mentioned. Things like canonicalization, making sure all molecule descriptions are standardized, very important for consistency. Also, consensus scoring. Instead of just relying on Vina, it averages scores from multiple docking programs like Vina and SMINA, makes the predictions more robust. And tier classification.
Tier classification sounds key for that shortlist. What does that involve?
It basically assigns the final candidates into tiers, tier 1, 2, or 3. This ranking is based on a mix of factors: how novel the chemical structure seems to be, the predicted binding affinity, and the strength of that multi-omics concordance data. Tier 1 candidates are your top prospects. Look novel, bind well theoretically, and hit a biologically validated target. That structured output is exactly what a pharma research committee would want to see.
And just to tie it back, the inventor, Ansari, even integrated his own symbolic system, these Zaban glyphs, symbolically encoding entropy collapse. That sounds unique.
It is quite unique. It reflects, I think, the highly multidisciplinary, maybe even philosophical approach needed to bridge Quantum physics, biology, chemistry, AI. It's his personal signature, a way to conceptually link the biological need with the quantum possibility within his framework. It shows it's not just a random collection of tools, it's a thought-out integrated system.
What's also really important, I think, is that this core idea using quantum entropy for better search and optimization isn't just for drugs. The sources mention QuantumLaso, LLC, applying it elsewhere.
That's right. They mentioned quantum tornado. It's a system trying to forecast severe weather, specifically tornadoes, earlier. The idea is to inject these quantum signatures, this true randomness, into atmospheric models. The hope is it can help overcome some of the deterministic blind spots in current chaotic weather simulations, potentially giving more warning time.
Fascinating. And also material science.
Yeah.
Energy.
Yeah, applying the same fundamental principle using quantum-seeded search to explore possibilities to finding new alloys, maybe better battery materials, things that standard deterministic algorithms might just overlook.
So the real essence of all this work, this deep dive, it seems to be proving that groundbreaking innovation doesn't always require, a seven-figure salary or huge VC funding. It needs curiosity, persistence, and maybe the skill to cleverly assemble these modern, powerful, and often accessible tools, from open source databases right through to quantum cloud platforms.
Absolutely. It fundamentally changes the narrative about who gets to innovate at the cutting edge.
And looking ahead, there's this concept of closing the loop, making the system learn from real-world results.
Yes, the active learning loop. That seems to be the next major evolution outlined. This is crucial. It means feeding the actual wet lab results. Did the drug work? Was it toxic? What was the measured binding affinity? Feeding all that back into the AI model.
So the system doesn't just run simulations, it learns from reality.
Exactly. It refines its search strategies based on what actually works. And critically, this loop allows for direct empirical comparison. How did the QRNG-seeded candidates actually perform in the lab compared to the PRNG-seeded ones? Does that theoretical novelty driven by quantum entropy translate into tangible, real-world advantages? That's the ultimate test.
That active learning loop ensures the system gets smarter over time, not just faster.
Right. So when you put it all together, the convergence of these powerful open source tools, readily available quantum entropy sources, sophisticated AI, it really does feel like the path to discovery is genuinely opening up. It's becoming more accessible for the determined individual, the small team. The traditional barriers are definitely lowering, which really makes you think, what boundaries will you try to push?